system here. So everyone at your seats, you should see like one sticky note, a yellow sticky note. So write your full name on it, and at the end of the talk, so like at the end of the third talk, we'll have these boxes out, one box per presenter, and you can put your voting slip into the box of the person you want to choose. So we're not going to make those available until the end of the talks, though. So we really want you to stay and watch all of them before you vote. Yeah. Oh, another note about C's Penn iTalks is that other schools, School of Arts and Sciences, School of Nursing, and SP2, which is social work, they also have their Penn iTalks. So all those emails that I've sent out to you guys, there's the Penn iTalks website. I highly encourage you to check those out too. They have similar events, but different format from this. But yeah, so thank you guys all for coming, and I'm going to hand it off to Ryan.
Okay, thank you everyone for coming. May already did all the introductions, so I guess I'm more like the flavor flav of this thing and just kind of hyping everyone up. So as she mentioned, the audience favorite when you vote will get $100 for presenting today, which I've never won anything for presenting anything, so that's pretty cool. And then on top of that, there'll be another round where we have another 3 speakers, and one of the 6 speakers in total, based on the judges voting, will win $500, which I'm pretty sure no one here, other than probably the 3 right there who've won many grants, have won $500 for their academic research. So that's a pretty cool thing, and on top of that, there'll be beer and a happy hour afterwards, which is probably the second reason you guys are all here. So without further ado, we'll get this started. So I'll introduce Ben Friedman, who's in his 3rd year in Lou Soslovsky's lab in BE. He had his BS from University of Rochester, and he'll be talking today on tendon fatigue injury and the mechanisms in novel mechanical and image-based measures. You can read the title just as well as I can. So I'm going to hand this over.
Well, I've always been fascinated by the human body, and it's hard to say when that spark was first lit. But the first photo evidence goes back to the 4th grade, when I built this human-like robot, taller than I was at the time, with a little bit of help from my dad, I'll admit. And this got me thinking, at a young age, how our bodies were put together and how we could do all the amazing things that we do. Our bodies are amazing structures, yet somehow we become injured. And these injuries don't just happen to athletes during sport, but to any of us. They can affect our shoulders, our ankles, and our knees. And really, these musculoskeletal diseases are highly prevalent. And as you can see here, they far exceed other incidences of other diseases, such as circulatory, diabetes, and respiratory. They decrease our productivity and ability to do normal living activities, and therefore also decrease our productivity. And you can see here, musculoskeletal diseases are leading the way. But there is hope. The good news is that Penn has many great orthopedic research facilities, labs, led by the Penn Center for Musculoskeletal Disorders and our very own McKay Orthopedic Labs in particular. This opens the doors for lots of biomechanics research opportunities, such as tendon biomechanics in the Soslovsky lab, and Achilles tendon in particular, which is the focus of my research. So what do we do in our lab? We look at a number of things. We look at tendon and ligament mechanics, how strong tissues are as we pull them apart. We look at things like image-based measures of tendon structure. We look at in vivo functional surrogates of tendon healing. We look at some of the biological mechanisms that govern healing as well. So before I go too far, I just want to talk about really what is a tendon in the first place. So tendons transmit force from muscle to bone, and they control movement, from some of our most explosive motions to some of our most finest. Tendons also have a hierarchical microstructure, and if we take a transverse cut at our tendon here, we can see it's composed of smaller pieces that originate from the large tendon scale and go down to the single molecular scale, down to a single collagen molecule. And if we look at this, the structure at a smaller scale, we look at, say, an SEM image, and this is submicron, we can see that the tendon is composed of these longitudinal fibers that are primarily oriented along the long axis of the tissue. However, if we look at an injured tendon, for example, we can see that it's much more wavy and the fibers are bent and jagged. And you can see that it's definitely much more disorganized than the picture on the left. And understanding the causes and effects of this phenomenon is an interest of our lab, and we believe that one of the underlying contributing factors is fatigue-induced damage. And we approach this question, like many questions in our lab, from both a structure and function perspective. And when we're talking about structure, we talk about things like the orientation, the topology, the alignment of these tissues. And by function, we are a biomechanics lab, so we can look at things like the material properties of tissues, and we can also evaluate things like gait, and in the case of the Achilles tendon, things like the range of motion of a joint and its stiffness. So tonight, I have two stories to tell. The first is on fatigue in the context of Achilles tendon healing. And the second is taking a little bit closer look at these tendons into a phenomenon known as tendon crimp, or the waviness of these fibers. So I'll start off by talking about Achilles tendon healing. And basically, the Achilles tendon ruptures go way, way back into the age of Greek mythology and Achilles himself, before there was such thing as an Achilles tendon rupture. And although although now we have a name for this type of injury, there's still a lot that we do not know. But we've learned a little bit about ruptures since. We know that the incidence of Achilles tendon ruptures is rising. We know that about 1 in 10,000 will suffer a rupture in North America each year. And this is a rupture, not just a sprain or a strain to the tissue. We know that the Achilles tendon has highly repetitive loads at or near failure during normal activity. And what that means is as you're running, you're skipping, you're hopping, the loads that your Achilles tendon might be experiencing are really not that far off from what it might take to pull that tendon completely apart. There's an interesting study that was done by the American Academy of Orthopedic Surgeons. They reviewed all the available literature on Achilles tendon injuries and treatments, and what they found is the result was that there was weak or inconclusive evidence for many treatments and rehab strategies. And this was interesting and kind of perhaps a shocker to the field. And our lab thinks that a major reason for this is that we haven't evaluated tendon mechanically as rigorously as we have in other materials, for example, metals or something like that. And historically, we've only evaluated tendon by taking it and we pull it apart. We do a ramp to failure. We see how, with the maximum forces, the thing that you learn about when you take your first mechanics class. However, for tendons like the Achilles that are subject to these repetitive loads, dynamic loading, at or near failure, we might want to think about providing a more rigorous method of testing. And if we want to really be able to determine if what the proper timing is for when an individual recovering from an injury may be able to return to their high-level activity, we should start to think about understanding healing in the context of fatigue loading. So when we subject tendon to repeated loading, we see a three-phase output for tissue stiffness and strain. And it looks something like this. And when we look at this, we see this three-phase output. Sorry, there's the input. Here's the output. For strain, we can see strain increases monotonically, and stiffness has this opposite response, where it increases a little bit, decreases gradually, and then drops as the tendon fails. And we think that this curve is interesting, but we don't really understand where it comes from. So we think that if we can understand how this curve is changing from both the structural changes and compositional changes with healing, that this may allow us to develop targets for potential therapies and enhance tendon monitoring. And if we can do this, we can perhaps provide an answer for when you can really return to activity. So the first study that I'm describing had 3 parts. The first thing we did is we made these injuries to our Achilles tendons. In mice, we put a little hole punch basically in the center of the tendon. And then we let the tendon heal for a period of time. And during this time, the mice usually watch HBO, they eat popcorn. They were watching the Olympics when we did this a couple years ago. And then we do mechanical and structural testing to see how strong these tissues are. So we evaluate fatigue mechanics with a novel mechanical testing setup with an integrated polarized light system. So basically what you're looking at here is there's the camera. Here's our little tendon. It's about 5 millimeters long. It's inside this water bath, and it's being pulled by an instrument, so a mechanical testing machine. And basically what you're looking at here are two polarizing sheets. So you could think of this, you could make one of these in your garage if you took a pair of your polarized sunglasses and you rip the lenses out or you pop them out. And then you orient them 90 degrees to each other. And then if you put a material like tendon in between them, the only way that light will get transmitted through is that there's some sort of a birefringent material that rotates the light. So that's what we're doing here. And as these polarizers rotate, what we see is basically changes in intensity of the tissue, which we can quantify as a metric of tendon structure and alignment. So basically the output from this is some measure of alignment and structure of the tissue. We also can look at mechanical properties as well. So this is looking at our tissue. We subject it to, again, our fatigue loading input, and I showed you this plot just a second ago. And if we look at what strain looks like, we see it basically mirrors what the force does. And if we just follow the peaks of this strain curve, we can quantify the peak strain shown here, and by quantifying basically the slopes of force and displacement, we can quantify how stiffness varies with time, generating this kind of plot. So I'm going to show you some results, and basically the results are separated into the different phases of fatigue life. So we can basically probe this, this fatigue response at 5%, 50%, and 95% of fatigue life. So what do we see? So at first glance, this is looking at stiffness of the tissue, the dynamic stiffness versus the percentage of fatigue life. And you can see basically healing here where we have controlled tissues, uninjured controls, and then how they heal over the course of 6 weeks. So at first glance, it might look like stiffness recovers with healing. But we see that if we induce fatigue loading longer, beyond 5%, we see that in fact it does not. And therefore, this dynamic stiffness property, as assessed throughout fatigue life, seems to provide a more rigorous standard of healing than might be observed if we just pulled that tendon to failure using conventional basic methods. So we looked at a number of other mechanical properties as well. In particular, we were interested in seeing how far we could push this tendon in an injured state before it could fail. How many cycles can you go before it fails? So we see this response. We see a dramatic effect here. So although we saw the stiffness in the previous slide drop by 25% following an injury, we saw a dramatic 37-fold drop between a control and a weak zero tissue that gradually recovers with healing. Interestingly, the pilot data that was conducted for this study showed that an injured tissue had roughly 60% of the failure load, and that's just using the conventional ramp to failure methods of the normal failure load. And this demonstrates the significance and importance of this measure over some traditional mechanical tests. Furthermore, we saw that structural properties assessed using our polarized light imaging system during healing predicted mechanical properties. So right here, what you're looking at is our structural measure of organization, which is simplified for the purposes of this talk, and versus the dynamic modulus, which you can think of as a measure of stiffness. And as you can see also that the tissue became more disorganized with healing as well. So we've been really excited by these results, and we think that they have several applications. Applications to help define rehabilitation strategies for when it may be safe for individuals recovering from Achilles tendon injuries and ruptures to return to activity, as well as in enhancing our current imaging modalities for evaluating tendon structure, using ultrasound, for example. We also think it has applications in tissue engineering. Our results have demonstrated the importance of fatigue loading in evaluating the mechanical integrity of these tissues that are subject to repeated loads, and for other tissue engineering constructs like an electrospun tendon or the meniscus or the heart, tissues that experience highly repetitive loads, that we think that this work also has applications there as well. So just to dig a little bit deeper on this topic, we've continued to ask, what is an injured tissue and how can we measure such an injury in vivo? And that brings us to our second story here on this concept of tendon waviness or crimp. So as I mentioned before, tendon's complex structure relates to its mechanical properties. And if we pull this tissue apart, if we just do a simple, pull this tissue to failure, instead of seeing that linear elastic response that some of you may be familiar with, if you pull the metal apart, we actually see something quite different. So this is looking at force versus displacement, and we pull it apart, we see this distinct nonlinearity or toe region present. And oftentimes that's attributed to the straightening of these fibers, the collagen fibers, or uncrimping as we call it. You can see they look straighter here than here. So we can measure this crimp phenomenon using our polarized light method, and you can see there's banding on this image in particular at low loads, but at higher loads this goes away. So you can think of this as the tendon being very wavy, and then just like you're tuning a guitar string, that it becomes more aligned. So in this second study, we asked if crimp indicates mechanical damage. In doing so, we subjected tissues to fatigue loading and then evaluated their mechanical and structural responses as a result. So that's what you see here. So again, we apply a fatigue loading input, and then we can look at our same mechanical responses, but ask, what can we see with this crimp phenomenon, and what is changing there? So basically, in terms of our methods, this is a little bit repetitive, but you can see that we basically subject our tissues to a fatigue loading input, and then we can do our image capture at different set loads that may be indicative of where tendon crimp starts to go away from what we know. And then we can see how the tissue looks initially, keep fatigue loading, and then we can see how it looks after a number of cycles. And then from these, we can quantify the response in different regions. There have been some previous studies that have indicated that tendon is not homogeneous, that the properties within a lot of these biological tissues actually vary quite amazingly. And that provides additional complexity with a lot of this work. So with this, we can quantify properties like our crimp frequency and crimp amplitude. So we found here, visually, you can see that the tissue on the right looks different than the tissue on the left. We see that fatigue loading increased the crimp amplitude and decreased the crimp frequency. And if we look regionally, we find that at low loads, our lateral, our side region of interest has an increased crimp amplitude and a decreased crimp frequency compared to the center. And we also find that at high loads, interestingly, these differences go away. So clearly there's a load dependence response. We also were able to relate the crimp amplitude to the measure of mechanical damage, and we see a strong relationship as well. So we think these findings have applications in enhancing the clinical translatability of image-based measures of tendon structure, such as ultrasound or MRI. In addition, the changes observed in tendon structure with fatigue loading provides insights into potential alterations in the extracellular matrix that cells may feel and respond to following high dynamic loading. So just in summary, this talk is focused on the concept of fatigue loading addressed from a well-controlled structure-function perspective to elucidate some of the mechanisms that may govern healing and injury in tendon. In doing so, we've really just started to scratch the surface for an exciting journey we have ahead in the field of tendon biomechanics. In these studies, we've shown that injury and healing affect tendon fatigue mechanics and structure, and that this phenomenon of tendon crimp relates to tissue damage. Currently, we're working on these topics in larger animal models to provide a comprehensive approach to study Achilles tendon healing. So just to come back to our original slide. So we're able to visualize strain fields in injured tendon to see how that might be changing with fatigue loading. We're also able to exercise some of our test subjects to try to really drive home the point of when it may be safe to return to activity. We're doing all sorts of image-based work to try to assess tendon structure using high-frequency ultrasound. And we're also able to look at how injured tendon looks, we're also able to look at the vessel architecture as a potential mechanism that may also drive changes in mechanics using micro-CT. We're able to look at different functional surrogates of in vivo tendon healing using basically gait data in animals, and also immobilize certain limbs to try to mimic the clinical situation. We're also looking at some of the biological mechanisms that govern the healing process. So we think we have an exciting road ahead as we continue to uncover some of the mysteries of Achilles tendon injury and eventually hopefully prevent these devastating injuries from occurring in the future. Thank you.
So we have time for a few questions.
Hi, Ben. That all sounds and looks very painful to me. But kind of thinking about your work and how it can be used to develop new therapeutics. So rather than only preventative or thinking about tissue engineering after complete rupture, are there therapeutics that can be used to alter the organization or the structure of the ECM in someone maybe who's somewhere in between that stage?
Sure. So the first therapeutic we're looking at right now in a larger animal model is the effects of just simple immobilization. So is there a simple exercise therapeutic that can potentially help these ruptures. So there's a lot of suggestions from the physical therapy world that early loading can be beneficial. So we're looking at that and returning our animals to activity earlier or versus keeping them in casts later. So we're also looking at some other things in terms of the effects of a surgical repair or not a surgical repair, which could also have an effect on some of the organization properties that you talk about. There have been a lot of biological studies that have been suggested using things like all sorts of different biological therapeutics like PRP, which some of you may be familiar with. However, these have been extremely controversial in terms of their actual benefits on the tendon healing process and organization. And many of these studies have actually not evaluated the mechanical properties. They may just simply look at just the histology, for example. So you don't really get a full flavor for that. So I think there's definitely work to be done in terms of potential therapeutics, but we don't really know the answer to that yet.
Okay, great talk. I just wanted to understand your data a little bit better.
Sure. So
in the first part, you showed this dynamic stiffness versus percentage of time to failure.
Yes.
And you said, oh, that doesn't recover on healing. But then your next graph sort of said, oh, the number of cycles to failure is dramatically less before you've healed and then gets longer. So doesn't that just say the dynamic stiffness isn't really telling you about the healing process?
Yes, that could exactly be telling us that it doesn't. So it demonstrates a couple of things. So number one, it doesn't show that we're able to show full recovery. In the physical therapy world, sometimes they'll try to think about stiffness and say, oh, that's really recovered. Or if you're doing a simple test to failure, like many groups do, they may say, oh, we've recovered, we're not recovering stiffness, therefore we're not really recovering, or vice versa. They may not really detect how big of an effect an injury might make on a tissue if we see the huge drop in cycles.
Because you don't want to cycle the patients to failure.
No, we don't.
Okay.
So that's why we do controllable animal model studies, because we'd love to do this in patients, but we can't really get them to give us their Achilles tendons.
And then
for a variety of reasons.
quick follow-up was, okay, you showed the birefringence actually was getting more disordered as it healed.
Yes.
Like, is there a quick answer for how I can understand that?
Sure. So very common that after an injury to a tendon, that it becomes more disorganized. So that's what we're seeing, and it's in agreement with that. At the same time, the cross-sectional area of the tissue is increasing. So that could be a potential mechanism that may be altering the effects. There could also be better collagen that's deposited. In addition, the effects that we showed with the increase in disorganization was evaluated throughout the entire tissue. It's possible that local, more regional changes may be occurring adjacent to the injury sites that could be showing more mechanical improvement.
Very nice work. It looks like, and maybe this is not important, but it looks like you're treating tendon as an elastic material. My question is there any possibility that these are viscoelastic?
They're absolutely visco.
And thus the rate there are a lot of rate dependencies that you haven't perhaps studied, or is that going to be important?
Sure. We measure viscoelastic properties with our dynamic test. So I didn't show you all the mechanical data, but we are looking at both elastic and viscoelastic properties. So I showed you the dynamic modulus, but we can also look at different fascias and things like that as well. So we're absolutely characterizing those properties. I just didn't show them today. Well, we do see viscoelastic changes as well with this and with a lot of our other additional tests and experiments going on in our lab looking at dynamic loading.
Time for one audience question. Don't be too shy.
Yeah, I was curious. It's a waviness rather than crimping. It's waviness, and once you extend it, then you start actually damaging the cells. And if you then let it recover and heal, won't it heal? Anything that's broken might heal and actually lock in that waviness of the cell. So would it be helpful to cycle it loosely through the healing process to encourage it to re-crimp?
That's a great point. So to make sure I'm understanding your question correctly, you're proposing that to enhance tendon healing, we might do some sort of an in vitro study where we try out different types of dynamic loading to try to improve crimp, for example.
Yeah.
Okay, so that's a great question. So measuring crimp in different frequencies and how that might affect crimp with an in vitro. So that's a great point. So we didn't look at that directly. So all the testing that we're doing is ex vivo from the animal for these fatigue tests. But you could imagine designing a study where you could take a tissue, put it in some sort of a bioreactor that could cycle, and be able to look to see if we're promoting good matrix production with various different types of cyclic inputs and different things like that. So there are some groups that are actually looking at those features and trying to link some sort of mechanical input response to biological gene expression, and they're doing that in live tissues.
All right, let's thank our speaker one more time. Okay, transitioning from bioengineering into material science engineering, we will have our next speaker, Carlos Aspetti, and he is currently a fifth-year PhD student in Dr. Ritesh Agarwal's group. He received his bachelor's degree and master's degree from Drexel University, and if we have time while he's still setting up, I have a fun fact that his favorite breakfast food is huevos rancheros. So I'll hand the floor over to Carlos.
Thank you. All right, so I'm going to start setting up. But I should let you know that I'm actually having knee surgery next week, so I should probably talk to Ben later. Okay. Testing, testing, 1, 2, 3, perfect. Can you guys hear me in the back? Yes. Great. Let's do this. Silicon photonics. Turn off the dark. As the title suggests, silicon, the backbone of modern electronics, is a dark material. It does not emit light. But by the end of this talk, I hope to convince you that by combining optical engineering with material science, we can challenge this fact. From electronics to photonics. You know the microchips in your cell phones and your laptops and your devices? They're based on the integrated circuit. The unit of logic of which is a transistor. It's what computes the zeros and the ones. And that's made of silicon. Silicon is mechanically robust, conductive, abundant, non-toxic. It's really a wonderful material. And the way these guys communicate with each other is by shooting electrons down a copper wire, exactly as you would expect. But electrons can only go so fast. A copper interconnect operates at about 10 gigs. Wouldn't we want to do this with light, with a photon? After all, light is the fastest thing in the universe. In fact, we can. So we already have optical interconnects operating at 100 gigs. But the issue here is that in order to interface your unit of logic with light, you require an external light source. Why? Because silicon is a dark material. It does not emit light. But then why not just build the whole thing out of optical semiconductors? Optical semiconductors abound. But the issue here is more of an economic one than a scientific one. Intel, AMD, Taiwan Semiconductor. The semiconductor industry has decades and billions of dollars of experience working with silicon, and they're not going to change. So to quote Mario Panicia from Intel Photonics Lab, the goal is to siliconize photonics. And the final piece of the puzzle, arguably the most annoying one, is the need for an all-silicon light source. So if you're driving around Philadelphia, you may have noticed these, or maybe you didn't. But if you did, they're made of light-emitting materials, light-emitting semiconductors. So in order to understand how a light-emitting semiconductor might work, might emit light, we're going to do an experiment together, right here, right now. So this is a light-emitting semiconductor, and I'm going to supply it some high energy. You can do this many ways, but I'm going to shoot it with a laser. And what's going to happen is that the electrons from the valence band are going to graduate to the conduction band, okay? High energy. But physics is lazy. We want to be in the lowest energy state possible. So this electron is going to quickly relax to the ground state, which is the conduction band minimum. It can then recombine with the hole that it left behind and make light. So the bioengineers in the audience can think about fluorescence. This is fluorescence from a semiconductor. No big deal. But then we have an issue of timescales. This relaxation process happens on the order of a picosecond, whereas this recombination process happens on the order of a nanosecond. It is 1,000 times slower. So now the chemical engineers in the audience can think about kinetics. The fast process always wins. It happens first. And if you're my nephew, you can think about the tortoise and the hare. Except that in the real world, the fast guy always wins. Not the good guy, the fast guy. This is why this is always red, always yellow, and always green. Materials emit from the ground state. But we still haven't answered the question as to why silicon is a dark material. And that's because I haven't given you the full picture. You see, a fundamental law of physics is that both momentum and energy and momentum must be conserved. So let's repeat that experiment, but from the point of view of energy and momentum. So here's that same electron, that guy over there, that guy over there. It is excited to the conduction band. And now it has two choices. It can either recombine immediately with the hole to make light, or it can relax, accumulate here at the ground state, and then recombine, and then emit light. But remember that the time scales here are completely different. So this guy, the recombination process, is 1,000 times slower, right? So before the electron can even think about recombining with that hole, it's already at the ground state. Now the key finding here in this picture is that the ground state and the conduction band and the valence band, they're at the same momentum. They are momentum matched, which means that momentum conservation has already been taken care of. Thus, this process is actually very efficient. The efficiency is about 100%. If you have 100 electrons, you're going to get about 100 photons out. You want to make light emitters out of, or rather your street lamps out of this stuff. But what about silicon? Well, silicon is actually very similar. Here's your excited electron, and it's going to quickly relax to the conduction band minimum, right? Picoseconds. But the most important fact here, this is probably the most important fact in the entire talk, is that the ground state here and the valence band are momentum mismatched. They are at different momenta, which means that in order for this process to happen, in order for the electron to recombine with the hole and emit light, we must exchange momentum with the surroundings, which is actually an extremely inefficient process. This happens about once in a million. 1 in a million. Silicon is an abysmal emitter of light. But I'm going to rephrase the problem. You know, in silicon, once the electron reaches the ground state, it's basically gone. So if you want to get light out of silicon, it behooves us to do so before the electron is gone, right? We want to make the electron and the hole recombine before the electron has time to relax. So the question I ask you is, or ask myself rather, can we make the electron and the hole recombine, and can we do that so fast such that we can actually compete with this time scale, with relaxation? Can we put that turtle on steroids? That's my question. So in order to answer this, we're going to go back to research from 1946. Like physics never dies. This is the heyday of radio operation. This is radio frequency research. And it turns out that the rate of radiative recombination, the rate of light emission from, for example, an electron-hole pair, is a function of your surroundings. It is directly proportional to the quality factor over the mode volume. What's that? The quality factor is the ability of your surroundings to store electromagnetic energy. The electrical engineers in the audience can think about a capacitor, right? It's also a measure of how long the light that's emitted actually sticks around. Does it stick around or does it just go into the vacuum? And in the denominator here we have the mode volume. And the mode volume is simply a measure of how tightly you can squeeze that light into a small area. You want a lot of light per area. So in other words, if your environment can potentially host a lot of light, then the light emission process, whatever that is, becomes more likely and therefore faster. And the way I like to think about it is like this. You know, if I'm playing golf, it's a lot easier for me to get that golf ball into an empty swimming pool than it is to get it into a little hole, right? The former is more probable. That's the point. Well, it turns out that the radio frequency antennas, the antennas on your phones, for example, do this stuff all the time, this system. An antenna takes, for example, a light that's on the order of a kilometer big, like a huge electromagnetic radiation, and works with it on the order of a meter, 1,000 times smaller. Radio antennas funnel electromagnetic waves into small spaces. Keep that in mind. But if you want to get visible light from silicon, we need an optical antenna, right? In order to do that, we're going to do two things. We're going to go nano, and we're going to exploit this very interesting phenomenon. It turns out that if you shine light on a metal surface, The electrons, those free electrons on the middle surface, can literally anchor the light down to the surface. The light becomes confined to that surface. It can only travel on the surface. This coupled system is called the surface plasmon, but I just call it nanophotonics. Metals allow us to do optics on the nanoscale. That's all. These metal balls are fantastic at doing this metal particles. And basically what's happening is you're taking a micron-sized radiation, or light waves, and working with them on the scale of a nanometer. Kilometer to meter, 1,000 times smaller. Micron to nanometer, 1,000 times smaller. These are optical antennas. Optical antennas funnel large electromagnetic waves into small spaces. And to drive the point home, here are suspensions of different sized gold balls. You get the colors of the rainbow. Why? Because these are different optical antennas. Slightly different size. Optical antennas. So can we use this process of enhancing light emission with an optical antenna to make light from a semiconductor faster? And we're going to start working with an optical material simply because it's easier to do, for now. This is the picture to remind you. So we did this the following way. We took an optical semiconductor here, it's cadmium sulfide, and we put our optical antenna around it, it's metal, and what we found was something fascinating. Once the semiconductor emits light, rather than having that light radiate into nothingness, it actually sticks around. It goes round and round the semiconductor. And it's highly confined, nanometer-sized dimensions. And we can actually take this computational simulation data to compute this enhancement factor. How much faster is our light? How much faster is our combination? The quality factor is pretty good, but I want to focus on the mode volume. The light in our system is 10,000 times more confined than it would be in free space. 10,000 times. We actually compute that the recombination rate here should be 1,000 times faster than it normally would be. It should be picosecond level. And when we first did this, no one believed us. It's too extreme. So we actually did some time-resolved fluorescence data, experiments rather, and we showed this experimentally. This process is now a picosecond. So in theory, we can now compete with the relaxation process and get light before it goes to the conduction band minimum. But like I mentioned before, an optical material already makes a lot of light, right? The efficiency is really high. It's 100%. So what do we care? Well, first of all, the turtle is now on steroids, so that's good. But in reality, we should think about silicon. And let me remind you, in silicon, once that electron goes to the conduction band minimum, it is long gone, right? So having this process become faster such that we can prevent this from happening to begin with is a good idea. And before we do this experiment, I want to know what the spectrum of silicon actually looks like. How much light does it emit? Okay, no help there. But we're going to try it anyway. So here is silicon, and it's interfaced with an optical antenna, metal antenna, as per usual, right? No big deal. We compute that potentially